I'm greatly honored to be invited to have the opportunity to participate in this symposium, and I thank Daniel Conley especially for all the arrangements for it. And also I'd like to thank the first two speakers for providing the introduction that I had prepared. You're going to have to sit through it again. The main thing I thought I might have to do is impress upon you the importance of nitrogen, but Andreas has already done that. And I'll give you my view of that anyway. And he's also explained that everything starts at the top, so everything that happens in the bottom of the ocean and anywhere below the euphotic zone depends on what happens in the euphotic zone because of the primary production is restricted to that region. And so everything is dictated from that, both vertically and in the idea of circulation of the ocean. I'm going to focus on oxygen minimum zones in the sort of the open ocean oxygen minimum zones, not the coastal zones. So this is our real focus on the ocean rather than coastal systems for the day. And mostly the reason that we care about oxygen minimum zones, and as I call them depleted zones, ODZs or OMZs, is because of what happens in the nitrogen cycle in those regions. And so that is the microbiology that I will focus on, the microbial communities that perform that nitrogen cycling in that region. And as Andreas mentioned, the loss of fixed nitrogen by denitrification and anammox balanced by nitrogen fixation is the reason that we care about the interesting, unusual nitrogen cycle of oxygen minimum zones. So I thought I would start with, in my naive attempt to convince you that nitrogen was important, why is the ocean blue? And the reason for that is that there's nothing absorbing the light. So there's not very much growing in vast parts of the ocean. This I took over the side of a ship on a research cruise in the Sargasso Sea. And it's really blue in the Sargasso and much greener closer to the coast because there's a lot of stuff growing there. So that's the primary production. And the reason for that lack of growth in the ocean is the lack of nitrogen for vast areas of the surfaced ocean. And of course, I now know that you all are aware of the limitation law, that it's mainly nitrogen limiting growth in ocean waters, and we understand that it's more likely to be phosphorus in lakes and ponds. And that means that if you do the experiment, you should be able to add nitrogen and get more growth and more accumulation of biomass. And that's an experiment that's been done in lakes. This is a picture of one of the experimental lakes in the Canadian system in which they added both carbon and nitrogen to half the lake and carbon, nitrogen plus phosphorus to the other half, and they got this dramatic demonstration that it was phosphorus that was limiting that system. And I had this slide in here before Wally Broker died this week. When he spoke at an ASLO meeting that was devoted mainly to talks about the Canadian experimental lake system, he stood up and said, well, I work in Lake 1, and that was the ocean. Well, it's a lot harder to do the experiment in the open ocean, and in fact, despite all the data on the deoxygenation and changes in oxygen concentration in the global ocean, it's still debated as to whether or not we have actually fertilized the ocean as a whole, in the open ocean, enough to see any effects on nitrogen cycling and productivity in the surface of the open ocean. But fundamentally, if nitrogen is limiting, then lack of oxygen is what keeps the ocean blue. And there is very little nitrate, as you can see from this map of surface nitrate concentration at the time when it is supposed to be the maximum. And in the minimum, there's vast surface areas of the ocean have zero nitrate, essentially. And even at the best of times, it's almost zero in vast areas of the surface ocean. And that means that that's where nitrogen is seriously limiting, that the productivity is restricted very much to the other parts of the ocean. Those are not very large pieces of ocean where most of the production happens. And we see that the highest production is associated with regions where nitrogen is present, but where nitrogen or iron are the limiting factors. So where that's supplied, you get more productivity. And those are the regions in the coastal and near coastal regions of the western boundary. Western edge of continents, eastern boundaries of oceans. And what that leads to is this productivity in the surface ocean, as you can see in these regions which we're going to focus on today. Photosynthesis and respiration we've just discussed, Most of the photosynthesis and respiration happens near the surface. That's where all the photosynthesis is, and most of the respiration associated with that also happens at the surface. But there is some material that's produced in the surface that makes it to the bottom of the ocean. And this is a much exaggerated picture of the material that's produced in the surface ocean and then sinks a very small proportion of it actually reaching to the bottom. And we measure that using sediment traps, so you're physically capturing the material to figure out how much is produced at the surface and how much makes it to depth. And this is the very famous Martin curve, which shows an average of several studies in which they quantified the amount that goes missing from what you catch in a bucket here, basically, to what you catch in a bucket here. And it's well established or well accepted sort of average number that 1 or 2% of the material that's actually produced up here makes it down to a few thousand meters. So that means it's being respired in between, and that's the consumption of oxygen that we just heard about. And when you run out of oxygen, then you turn to nitrate. So this is a profile of oxygen through one of those areas where you have the really high productivity in the surface water and the coastal upwelling, or the oceanic upwelling as well, and you deplete the oxygen really to zero. There was a big debate for a number of years over whether it was really zero, but now we really know that it really is zero. It's not just a little bit. And it's a zero in some parts of the ocean for hundreds of meters. So that's important because that means that oxygen cannot diffuse to 400 meters from 100 meters. So it's really anoxic for a long time. And so that means that this curve also occurs in oxygen minimum zones, but it must be due to anoxic respiration. And that leads to this consumption of oxygen leads to this picture which Andreas already showed you of where the oxygen minimum zones are in the global ocean. And these are much exaggerated because I think this is the 20 micromolar limit, which is really not anoxic, but it's really low oxygen. So these are the places where this fundamental, interesting anoxic nitrogen cycle takes place. And here's where it's important to introduce the difference between fixed and biologically available nitrogen. There's a lot more in the atmosphere and ocean. This should be orders of magnitude bigger than this circle than there is fixed nitrogen, and that's the nitrogen that's limiting for primary production. And the exchange between those two pools, as Andreas mentioned, is that you add fixed nitrogen by nitrogen fixation, and you remove it by denitrification and anammox. And since the ratios of the removal depend upon the composition of the organic matter, it all depends upon denitrification producing the substrates for anammox. So that's why we can all be excused for being lazy and just saying denitrification, because the whole in-loss process depends upon the input of organic matter, and anammox can't deal with organic matter. It's an autotrophic process, so it requires the N2 substrates to be produced by denitrification. Both denitrification and anammox, though, occur only in the absence of oxygen. And so that's why the in-loss in the global ocean inventory depends upon these actually fairly small regions of oxygen-depleted zones where the oxygen is low enough to induce anaerobic respiration. And that leads to the measurable depletion of fixed nitrogen in these regions that underlie the oxygen minimum zone. So that's where N star, which is the net loss of fixed nitrogen, is actually greatest. So the highest negative numbers are in these oxygen minimum zones. So that was all to establish that nitrogen limits production in vast regions of the world ocean. The sinking matter that's organic matter that's produced in the surface is decomposed at depth, and that leads to the oxygen depletion, such that fixed nitrogen is removed under those anoxic conditions in ODZs and in anoxic sediments in the hemipelagic sediments of the world ocean. And that means that the microbial biogeochemistry of low oxygen environments controls the nitrogen inventory of the ocean. And so in an oxygen minimum zone, we see this characteristic chemical distributions. And you saw this was one of my slides that Denise showed, the dramatic oxygen minimum zone in the eastern tropical North Pacific. This leads to characteristic production of nitrite, two big nitrite maxima, one which is in the really depleted oxygen region, and this big maximum in nitrous oxide. And when you see these characteristic chemical distributions, you know you're in an oxygen minimum zone, and they imply the microbial control of the biogeochemical cycles that produce and consume these. And so my job here was to talk about the microbial communities that control biogeochemical cycles of oxygen minimum zones. I'm going to work my way from the surface down, and I'm going to stop here because then it sort of reverses and goes back in terms of oxygen conditions. So I'll start with the surface in talking about the microbes and the particular reactions that they control. So in the surface, it's primary production, as we've just discussed, and we'll see this figure a couple of additional times, so no need to memorize it now. So the photosynthesis in the surface regions of the upwelling zones is often big things like diatoms, and they are incredibly important. We think of them as being large cells, but I think that they're really sort of medium-sized cells that grow really fast, but they're large as far as grazers are concerned, right? So they're small in terms of being able to take up nitrate at a really high rate and grow really rapidly because individual cells are relatively small, but they're too big to be eaten by a lot of grazers, so that's why they grow to such huge, accumulate biomass and actually contribute a lot to export flux as well. So they're the ones doing this sort of thing in the very surface layer. But what we're most interested in is the next layer, and everything in an oxygen minimum zone, the most exciting, interesting, busy region is the upper oxycline and the top of the ODZ, and those regions overlap. And we have both photosynthesis occurring in that region and then processes that consume oxygen and take over and do metabolisms that don't require oxygen. So this is a picture of the oxycline. So this is a measured oxygen concentration, decreasing rapidly with depth. You always draw it as a cartoon as being smooth, but it's not. It's much more interesting than that. And then below about here, although this is a CTD, a seabird cast, we also had a stock sensor, which really gets down to 0, and it goes to about 0 right in there. So what was really interesting about this operation is that there are two chlorophyll maxima. So this green line is fluorescence, which is a proxy for chlorophyll concentration. And there's one big maximum in the upper oxycline, right where oxygen is decreasing, but it's the one that's responsible for the main primary production that's occurring here. But there's another one that's down here basically in the dark in the anoxic ocean. So that's the surface chlorophyll maximum and what we call the deep chlorophyll maximum, and they're due to two completely different assemblages of microbes. So it may be a lot of diatoms up here, but it's also these little red guys, and these are, I hope you can see that that's really red and really green. And these are cells that were collected between 0.4 and 2 micron filters with a McLean pump, so that's about 300 liters worth of the little tiny guys. And so it's Synechococcus that's present in the upper one, and Prochlorococcus that's present in the deep chlorophyll maximum. And so Prochlorococcus really does live in this dark anoxic ocean, slice of the ocean. But they are photosynthesizing. This really nice paper by Emilio et al., Emilio Garcia Robledo et al., showed that the Prochlorococcus in that deep chlorophyll maximum did respond to increased light with increased production of oxygen and increased fixation of carbon. The actual levels of light at the depth from which his samples were collected was somewhere between 0 and 10, so even lower than this. But this is encouraging. It shows that the cells are live there and they are doing photosynthesis, and they can do it at that really low, really almost dark level of light. But you don't measure any oxygen there. It's really 0. So that implies that there's a cryptic oxygen cycle. There's something else taking it up. And so that probably supports the other things that are going on in that layer, most important of which is nitrification, which is the oxidation of ammonium to nitrite and then nitrite to nitrate. And although we know that there are bacteria that can go all the way from ammonium to nitrate, there's no evidence so far that they're in the ocean, and we keep looking. So that's usually considered to be two different groups of organisms, the ammonia-oxidizing archaea. When you get to show pictures about this kind of biological organism, it's just not that exciting, you know. So little, you know, hot dog-shaped jobbies or little spiny things or little round jobbies, but these really are tiny. The AOA are on the order of less than a micron. Nitrospina is long and spiny, not quite as tiny. We think that's the major bacterial species that's important in oxygen-depleted zones, although there are more than one. And both of these processes require molecular oxygen. Actually, this one requires molecular oxygen in this process. This is requires oxygen only in respiration, so molecular oxygen is not required for nitrite oxidation. But every known nitrite oxidizing bacterium is an obligate aerobe. So they should be occurring and doing their work in water in which oxygen is present. But that's not what we find. In both of those major oxygen depleted zones, oxygen minimum zones in the North and South Pacific, this is a depth profile. It's labeled with density, but it's basically a spread out depth profile. For both of these regions, ammonium oxidation peaks right around the top of the region where oxygen becomes 0, which is below the dotted line. But nitrite oxidation is high, way below that. And so we think it occurs. We've been arguing for years that it really does occur in the total absence of oxygen, and I think we're about to figure that out. But both of these occur in that region between where Prochlorococcus is present and much deeper, where there isn't much oxygen. So we've been trying to chase that anaerobic nitrite oxidizer, which is a contradiction of terms, for some time, and recently using metagenomics have identified at least 2, and now more recently several more, MAGs, metagenome assembled genomes. It's not a political statement. And so far, these 2 are shown by assembling a total metagenome and have found 2 distinct nitrite oxidizers that are different enough from Nitrospina that they're not the same genus. So it's something we don't have in culture, we don't know much about except from these genomes. And this is their relative distribution in the metagenomes of the Eastern Tropical North and South Pacific, and also the Arabian Sea. And this gray box is where there is no oxygen. So in the Arabian Sea, this was actually a sample that was a station that was sampled just outside the major ODZ. So it's not really anoxic. But what you see is that of these 2 MAGs, one of them, the orange one, is really adapted apparently to the top of the ODZ, but the other one is relatively more abundant in the truly anoxic water. So I think that's a really interesting genome or microbe to go after to explain that. So a completely different approach to figuring out what's going on in the nitrogen cycle here. In other words, do we really have anoxic nitrate oxidation, is the use of the natural abundant stable isotopes distributions. This is with our colleagues, Karen Casati and her student at the time, Brian Peters. And their model, taking into account both our measurements of concentrations, isotopes, and rates, simulates, these are the model simulation, high rates of nitrite oxidation, about on the same order that we actually measure, and also of nitrate reduction, so the opposite process, also on the rates that we actually measure. So we usually think of nitrate reduction as being the first step of the complete process of denitrification. Oh dear, what did I do? Goes all the way to N2. That was lucky. But what we have evidence for here, it looks like, is that these 2 processes are just kind of spinning around in a futile cycle. So the high nitrite oxidation, high nitrate reduction, what is that doing for anybody? Before I try to answer that, there is other evidence for a lot of nitrate reduction. And this was a really exciting paper from Frank Stewart's group, in which he showed that there was high abundance of the particular genes that encode the nitrate to nitrite reduction step. So all of these colored bars represent genes that are present in SAR11 clades that contain nitrate reductase, and it's basically everything on the plot. So this is both in the ETSP and the ETNP. The DNA is very abundant, relatively abundant, in the oxygen minimum zones where there is no oxygen. And it was not known prior to this that any of the SAR11 clades actually did denitrification, or even the first step of it. And their transcripts are also present. So it looks like there's great capability for that reverse, the nitrate reduction. And just to remind you that SAR11 is the most abundant bacterium in the surface ocean. And so it's kind of interesting that it's also really abundant in the oxygen minimum zones and probably doing this nitrate reduction step. It's also tiny, tiny, 0.7 microns compared to a bead there. So what's happening with that cycle? We have evidence that nitrate is being rapidly reduced and recycled by nitrite oxidation in water that allow both of those processes to happen. This one is usually obligately anaerobic. This one is usually obligately aerobic. So what accompanies that, the nitrate reduction, is accomplished, in this case, if it's SAR11, and by sort of normal denitrifiers as well. They decompose organic matter and remineralize it to ammonium, in part, whereas the nitrite oxidizers are autotrophic, fix CO2, and make it back into organic matter. This is going to be important to the rest of the nitrogen cycle when we get there in a moment. So I think we're going to transition to the next layer, which is the core of the ODZ, which actually is disproportionate here. It takes up several hundred meters. And the primary thing that goes on in there is denitrification and anammox. And the microbes involved in that are even more boring. There are many, many hundreds, if not thousands, of different kinds of bacteria that are capable of denitrification, or at least of one step in it. And, you know, you don't see that in the morphology at all, which is one of the reasons that we've really been in the dark about how much diversity there was both in the denitrifying bacteria and in how the various steps of the pathway are accomplished. And that's really the opposite for anammox. I did not just say that anammox is boring, but in terms of the microbes that carry it out, there are 5 known genera, and only one is ever found in the ocean. And there's a tiny bit of diversity in that one genus in all the genes that we've looked at. So it's basically one thing. Quite different chemistry. This is an autotroph, again, fixes CO2, but the net effect of both denitrification and anammox is the loss of fixed nitrogen. And this one is dependent upon this one usually because of the necessity to break down organic carbon to get to the substrates for anammox. So we've done a lot of work on that, and I'm going to show you one whole slide on for data on denitrification and anammox, just to tie it back to the overarching model of how the ocean works, which is this supply of organic matter to depth dictates the shape of every other reaction curve with depth. And so the overall rates of denitrification and anammox now added together to give you the total N loss rate shows something that can be modeled exactly as a Martin curve. And that makes total sense, right? Because the material that is being decomposed by denitrification and therefore supporting both denitrification and anammox depends on what happens in the surface. So when you look at the rest of that model, you see that we also get anammox and denitrification approximately correct. They're almost on the same, about the same rate in this case. But the rates of transformation of nitrite and nitrate are much faster than total denitrification. So that's more evidence for that sort of feudal cycle that is going around much faster than the net transfer of nitrogen into the loss processes. And so we have anammox and complete denitrification supported by these intermediates, leading to the loss of fixed nitrogen. And that just reinforces that denitrification is probably a modular process. For a long time, we've thought about it as being one bacterium or one group of bacteria that take nitrate all the way to N2. But in fact, any combination is possible. And that leads me to the last step in the nitrogen cycle I want to talk about, which is involving nitrous oxide, which is an important intermediate in the complete denitrification pathway. And it shows that characteristic peak of concentration near the upper edge of the oxygen minimum zone, or the really depleted water here. So there's our nitrous oxide peak. And we've measured both the production and consumption of nitrous oxide to try and figure out what can maintain that peak. And what we find, using multiple different sources of substrate for the production of nitrous oxide, is that the highest production occurs right around where that peak is. And it comes from both ammonium oxidation, so nitrification as a side product of nitrification, and also from denitrification. But importantly, in the oxygen depleted zone areas of the ocean, the rate of production via denitrification is much, much faster, like 100 times faster than from nitrification. So nitrification happens all over the surface ocean. You multiply that number by the surface ocean number, and you get a lot of nitrous oxide. You multiply this number by a tiny part of the surface ocean, and you get about the same number. So it's kind of an important process, even though it happens in a really small place. And we've also measured its consumption, and this was a quick model that Andrew Babin did, combining measurements of consumption, modeling production. And the important thing is that right in this region at the top of the ODZ, you see a net production of nitrous oxide. And that could be shallow enough to be diffusing into the surface ocean, but more importantly, it probably shoals to the more surface regions and is sort of ventilated out. So the important thing is that it happens close enough to the surface and close enough to exit routes in the circulation that it constitutes an atmospheric flux. And the highest, once we put in our reformulation of the sensitivity of nitrification and denitrification to oxygen, and replace that into the model, Parv Centralinganum's model, you see the real hot spots of into flux from the ocean to the atmosphere are in those regions we've just been talking about, where we have the high anoxic nitrogen cycling and this production of nitrous oxide due to denitrification. So to put that into the future, which is not something that anything I've told you previously or anything that I've really worked on can inform, it's important to remember, though, that everything starts at the top. So just thinking very simplistically about how that affects the net production and possibly the flux of nitrous oxide, for example. If that's our model, if that's our sort of average N-loss rate, then that gives you an oxygen minimum zone like that. And how would that compare to an even sharper N-loss rate, which might occur with higher primary productivity? That would presumably give you a sharper and perhaps larger oxygen minimum zone, more productivity, more anaerobic respiration. And if you had less, then that would give you a smaller oxygen minimum zone. And you see that as you go offshore. For example, you see exactly this gradient if you go offshore from Peru or Mexico. And that would translate into then our normal sort of nitrous oxide profile, a higher and larger nitrous oxide peak if you had higher productivity, and smaller and lower nitrous oxide, and presumably accompanying differences in nitrous oxide flux. So of course, nitrous oxide is a really powerful greenhouse gas and ozone destroyer, so even just a little bit of it is something that is worth being concerned about. And that all relates then to the distribution of oxygen, just refreshing your mind everything that Andreas just said. Right. So this led me to try and think about what would be the net effect of global warming and deoxygenation on the flux of nitrous oxide and production in the world ocean. And it leads to a lot of if-thens, and uncertainty. So you start with the fact that nitrogen limits primary production, but you get high production in those upwelling zones because nitrogen is more available. That leads to an increased flux of organic matter to depth, which uses up the oxygen because it's limited by ventilation supply. That leads to anaerobic respiration using nitrate, and therefore you lose nitrogen. And by the way, get this production of an emission of nitrous oxide, which could lead to increased global warming, which leads to increased stratification and lower nutrient supply and more nitrogen limitation. So if you have decreased oxygen supply due to ventilation, would that lead to more nitrogen loss and more nitrous oxide? I think what you just heard from Andreas was that we really don't know, because there are really complex feedbacks and interactions that leads to a lot of scientific uncertainty. What I do know, though, is that the microbes are basically in charge. Now, Andreas said it was solubility and circulation, but in terms of the biogeochemistry, these distributions are contributed to, in addition to the physics, by the microbes, and I think that's where we need to focus some understanding on what maintains these distributions and what they mean about exchanges with the atmosphere. And with that, I think I just want to thank the main people in my lab who contributed the primary data, which I just showed you. Jimmy G was a graduate student. Shin Sun is the metagenomics person currently. Andrew Babin did some of the denitrification work, is now at MIT. Amal Jaikumar is my colleague at Princeton. Nick Peng was a student at the time who did the nitrification work I showed you. Thank you.